Hello everyone, I'm Torbjorn Ekrem. I'm a professor of biosystematics at the NTNU University Museum in Trondheim, Norway, and have been coordinating the Norwegian Barcode of Life initiative for a little bit more than a decade. I've been building reference library for quite some time, and now I'm involved in helping out with sampling in the work package for sampling in the Biodiversity Genomics Europe project that recently was launched. And today I'm going to talk a little bit about the challenging we see when sampling for barcode reference libraries in general in Europe. And we'll focus my talk on a number of different aspects: quality, DNA, standards and gaps. And I hope that it makes sense. A couple of years ago we did this exercise as part of the DNA Aquanet, a cost action, with a number of participating nations. Many of you are probably attending this conference. And in this study we looked at the gaps in the barcode reference libraries for aquatic organisms often used in biomonitoring in Europe. And we focused on aquatic organisms because this was the cost action was all about. But we included marine taxa, let's say, in the AMBI list. And we looked at marine fish, for instance. We looked at diatoms and a few different types of freshwater organisms. What we found in this study was that there are considerable gaps for many organism groups. Here's an example from the AMBI marine invertebrates list, where we see that in the case where we are happy with one barcode reference or more, there is a rather high coverage for decapods and a couple of other rather species-poor taxa. If we include GenBank, the private taxa in bold, and the public data in bold. But if we require at least 5 barcode references for each species in order for this to be valid, we see that the coverage decreases considerably for all groups, and in some of these groups all down to about 30 to 40%, which is really low for many of these groups, especially the species-rich ones here, and the arthropods and the annelids. So obviously there are still gaps in the barcode reference library. The question then remains, well, does it matter? Obviously, if we have a species list that we want to use for biomonitoring, any gap from that species list would matter. And a couple of years ago, or actually it's 15 years ago now, we did a first exercise when we wanted to see if we could assign, let's say, an approximate taxonomic rank to an unknown sequence, even if it's not in the barcode library. And we did this with the chironomids as a non-biting midges as an example. And here we see from this very small data set that genera are not monophyletic, and the distance between species and the tips here to the next species are very large. This means that we cannot use DNA barcodes that are unknowns, that don't have a match in the barcode reference library, and give them an approximate, let's say, genus name or a name of a higher rank than species level. So we need to populate the reference database with a lot of data. But this was a very small data set many years ago. So what happens if we do this on a larger data set? So I tried this some time ago using about 20,000 records from bold, almost 9,000 sequences, were larger than 600 base pairs, and looking at almost 700 species and 1,200 bins. In a neighbor joining tree, this resulted in about 101 PDF pages, and we won't go through all of those, of course, in this presentation, but just giving a snapshot of that, showing that the same situation still exists. There is a very long distance between most species of chironomids in the dataset, and it's so long that the branch lengths down, looking at the relationships between species, become short and uncertain, and the taxa, like Tanitarsus here for instance, are really not monophyletic at all. So when we had unknown sequences in this dataset, we cannot really tell which genus it belongs to. We cannot even tell which tribe it belongs to, since they are not monophyletic either. So we end up at a subfamily or maybe even a family level in some cases for the taxonomic assignment based on molecular or barcodes. Here's another one. Could only go to subfamily Chironomini. So filling gaps in the barcode reference library clearly matters. And in order to first of all find the gaps in the Biodiversity Genomics Europe project, we want to focus our efforts on pollinators and the terrestrial, freshwater and marine fauna that is used in biomonitoring throughout Europe, including also species that are invasive or doorknocker species for some reason in some regions. In order to fill those gaps, we have a few different sampling strategies, and we have about 15,000 species that we want to sample for. Both terrestrial field sampling and aquatic field sampling will focus on finding fresh specimens because they have a high DNA quality. They have a rather comparable, rather low sequencing costs. It can involve citizens' activities in bioblitzes and having NGOs sampling for invasive species, for instance. And it's a very attractive way to sample species when there are many specimens or many species that are lacking in the reference library from the same region. And we see this for many of the hotspot regions, especially down in Southeast Europe, Southern and Eastern Europe. But for many of the species then that are rare or that we cannot really easily find in the field, we have museum sampling as an alternative. And in museum sampling, we have a great advantage that we have access to type material, we have access to a bunch of rare species. They are very easily available, already sitting there, ready preserved. You don't have to spend time or money on going out collecting them, although that is a nice activity, of course. But we have some issues with the DNA quality, which is also almost always rather fragmented when the specimens are more than 10-15 years old. So for these species, we would need to use other sequencing techniques than Sanger sequencing, and in particular we want to focus on genome skimming in order to solve these issues. Many years ago, when the Barcode of Life initiative really started, the Consortium for the Barcode of Life generated a set of quality assurance measures that some standards that should be met in order to call something a DNA barcode. Now that was very focused on the sequence quality, and quite little was focused on the voucher and metadata quality. So a couple of years, or actually last year, this group of colleagues from the DNA Aquanet network published this paper on metadata standards and guidelines for specimen and DNA curation when we build the barcode reference library for aquatic life. And in here we focus on the importance of storage of voucher specimens so that you can in the future borrow these voucher specimens, look at their identifications, re-identify them as new revisions are published, for instance. And we mention also how different organisms require different ways of DNA isolation and DNA harvesting, and how that should be standardized and mentioned in the reference library. Also where the stored DNA is available and how that can be accessed by other researchers and used for other research projects. And of course a bunch of standards and guidelines for the metadata. For instance, everything belonging to and describing the specimen, the taxonomic information, the identification, who did it, when was it done, the vouchers where they are available and who can borrow them, and so forth, and also on which DNA markers have been retrieved from the same specimens. So all of these things are really important when building a barcode reference library. And in Biodiversity Genomics Europe, we are focused quite a bit on both the storage of the voucher specimens, the way we harvest the DNA. We have a special task that is particularly concentrating on metadata development, standardization, and also offering a way to store the DNA so that it is available for future studies. When we have the reference library and have developed the reference library, we have kind of considered all of these quality assurance and quality control issues up here. But we have to remember that when it is still when it is there and is developed, it still needs maintenance by curating the library entries and update the taxonomic backbone as this is being continuously revised through the dynamics of a taxonomy. And then, of course, there are quality assurance and quality control points for the utilization part of a reference library too, mostly connected to how specimens are being, or sequences are being made available through the DOIs and the IDs, and how the metadata output can be organized. And Biodiversity Genomics Europe want to look at all of these different aspects of the reference library building, maintenance, and utilization. So one way of doing quality control could be to compare taxonomic assignments made based on morphology with those made based on genetic similarity. Either by comparing, let's say, a barcode phylogeny with a phylogeny based on other molecular markers, or looking at the OTU clusters or bins as species hypotheses. So the picture here shows a bin page for chironomid that shows something about the average genetic distance and maximum genetic distance within that bin, and also the distance to the nearest neighbor. And by using that information combined with the taxonomic assignment, it's possible to see if the taxonomic assignments are concordant with the genetic clusterings or the bins in bold. And in order to automate this a little bit, some colleagues of mine, João Fontes, Pedro Tumal Pedros, and Felipe in Portugal, developed this BAGS tool, which is an R-based tool that does an automated barcode audit and grade system for reference libraries. In this tool, we can use a library selection, so a basic checklist. It can be filtered for marine taxa, and we can do a data mining in bold to retrieve sequence information from all of that taxa on the list. And what the software would do then is to check the bin clustering and the species name congruence and grade that congruence on a scale from A to E, where A is the best, where there is no conflict and we have multiple sequences showing the same pattern, and where E there is a big conflict between the taxonomic assignments and the genetic clustering. This way we can focus our efforts on the taxa and the genetic groups where that are graded D or E, and in that way improve the reference database through the curation of specimens and data records. For the future, one might look a little bit further for reference libraries. For instance, including multiple markers would be important for many different organism groups. Maybe through genome skimming, and also including symbiome information to reveal species interactions, either through their microbiomes, through parasites or hyperparasites, or fungi and other organisms within organisms. If we have a better knowledge of this connection and these interactions between species, we would also be better suited to do proper nature management of those species, knowing what happens if you remove one or more parts of this complex network. So as a summary of this talk, I would like to say that the gaps in the reference library really are important, and we need to fill them as much as we can in order to make a barcode reference library and DNA barcoding useful for biological monitoring. If we have diverse sampling strategies, both including fresh field sampling and museum sampling, and complement those strategies with the required sequencing technologies, that really will help us fill those gaps in the reference library. And also quality assurance and quality control really is crucial in order to keep the reference library up to date. And finally, I would like also to mention that it is important to make reference libraries available and accessible according to the FAIR principles. If we don't do that, the reference libraries and an analysis based on those reference libraries are not really repeatable, and can be compared across larger geographic regions, for instance, or between research groups. So thank you everyone for your attention. These are some of the partners that we had building reference libraries, and I will be happy to take any questions when we're in the panel. Thank you.